Perfecto. A ver. Muy bien. ¿Estamos entonces?
Sí.
Estamos dentro, ¿no?
Sí.
Sí, muy bien. Bueno, pues empezamos. Welcome to this seminar of our cycle of scientific seminars of the Instituto Rocasolano. As you all know, today our invited speaker is Dr. Rubén Hervás Millán, who actually is working at the University of Hong Kong. So, Rubén, good afternoon. It's 7 hours. So it's 7 in the afternoon. So just a few words about the professional career of Dr. Rubén, which is a very young and brilliant scientist. Well, in his predoctoral stage, he was working at the Cajal Institute here in Madrid, and he got his PhD degree in 2013 from the Universidad Autónoma de Madrid, where he studied before. Then he went to his first postdoc stay at the United States, in particular to the Stowers Institute for Medical Research in Kansas City, Missouri, from 2016 to 2020. That very same year, he was a visiting researcher at the LMB at Cambridge. And then he moved again to the Stower Institute until last year, in particular in March 2021, when he went to the Hong Kong University. And currently his present academic position is assistant professor at the School of Biomedical Sciences. Well, his research has been focused on the structural aspects of functional amyloids extracted from biological sources. In particular, he has contributed to define the first case of a prion-like domain that allows an enzyme to gain functionality via amyloid-like assembly. And this extraordinary work was published in high impact journals such as Cell. Another highlight in his career was the characterization of the function and structure of the aggregated state, isolated in this case from the brain of Drosophila, of the synaptic protein synthesis regulator, which plays a key role, a causal role, in transforming short-term memory to long-lived memory. And these results were published in 2020 in Science. So thank you very much, Rubén, for participating in this cycle of seminars. And go ahead, it's your turn. Thank you.
Okay, thank you very much for the introduction. And well, good afternoon. First, I would like to thank the organizers for inviting me to this series of seminars at the Rocasolano Institute. I have good collaborators and friends over there, so it is always a pleasure to come back, virtually at least. So today I'm going to summarize our main research interest, and in particular, I will be focused on the role of amyloids as a physical substrate of memory persistence. I don't need to introduce protein folding to this audience, but briefly, in nature, proteins fold very quick, in order of milliseconds or seconds into very complicated structures to do their jobs. And they do it because in most of the cases, they have a landscape funnel that drives the folding process from the unfolded state, here represented in the top of the funnel, to a minimum energetic state, which usually corresponds to a unique native and functional state. However, in our lab, we are interested in a particular group of proteins, which are called prion-like proteins, which can coexist in two different conformational states, like a monomeric state and an aggregated state. And sometimes each conformational state performs different functions. A property of these prion-like proteins is that they form a self-sustaining system. So once the aggregated state is engaged, it promotes the conformational conversion of monomers, creating this self-sustaining system. And normally the acquisition of this self-sustaining state is driven by a modular domain in the protein, which is the prion-like domain. And the aggregated state can have different nature and different stability, ranging from dynamic states like liquid or hydrogel states to static entities like amyloids or non-amyloid assembles that with different structural features. For example, amyloids are elongated protein aggregates, but when you look in detail, they are characterized by a cross-beta sheet fold where beta strands running perpendicular to the main axis of the filament, and there is an inter-strand distance of 4.7 angstrom between layers. So in this example, this is every layer corresponds to one single tau monomer, which is stacked along the major axis of the filament. And there is another class of stable aggregates, which is the non-amyloid class, that although they also can form elongated filaments, they don't have the specific structural features that I mentioned for the amyloids. So although normally they are associated with disease, prion-like proteins are also involved in diverse physiological processes in different organisms. They can function as storage systems, scaffolding platforms, or playing roles in the immune response, like the RIP-K1, RIP-K3, which play a role in the necrotic signaling in mammals, and which structure has been recently solved by Miguel Mompean and collaborators. So prion-like proteins can be implicated in diverse physiological processes. Indeed, computational analysis reveal that prion-like proteins are abundant in the proteome of higher eukaryotic organisms. However, it remains unclear what is the functional relevance of the prion-like features, if any. So today, there are just few examples of functional prion-like proteins in higher eukaryotic organisms. So in order to get insight into this question, we perform a systematic screen for prion-like domains on the whole proteome of Drosophila melanogaster. So here, we analyze the prion-like score, disorder score, and low complexity score in silico, and together with biochemical and biophysical approaches that I don't show here, we have finally selected 5 potential prion-like proteins in Drosophila that we are currently characterizing in the lab. So these 5 candidates are implicated in very different biological processes. For example, HR4 is an steroid hormone receptor involved in development. Murasca is an ubiquitin A3 ligase involved in memory. And then we have another 2 prion-like proteins that form functional amyloids implicated in animal development. The first example is Herzog. And we recently published that Herzog, although it is expressed through the embryonic development, it is a monomer with the diffuse localization on the cell membrane between developmental stages 1 and 5. but it forms amyloid-like aggregates on the cell membrane, specifically during gastrulation, which corresponds to the stage 6 and 7. And while the monomeric Herzog is catalytically inactive, Herzog amyloid leads to the gain of phosphatase activity, which is required for the proper establishment of segment polarity in embryos. So now, in collaboration with Kaushik-C and Javier Oroz, we are performing the high-resolution structural characterization of Herzog, both the monomer and the aggregated state from embryos, to understand what conformational changes are necessary to acquire this new phosphatase activity, which is important in the fly embryo development. The second example of functional amyloid implicated in development is pipsqueak, which is a transcription regulator. So what we have discovered, and I summarize in this slide, is that pipsqueak is an inactive monomer. It's an inactive in the state, in the monomeric state, but during early embryo development, pipsqueak aggregates in the Drosophila embryo, and these aggregates resemble an amyloid state. So we have developed a protocol to isolate the aggregated state of pipsqueak directly from the developing embryo. And this is how it looks like under an electron microscope. And this aggregated state recruits the polycomb group complexes, which is a transcriptional repressor complex. And it does it more efficiently than in the monomeric state. As I show here in these plots, polycomb alone, it is elutes at the molecular weight volume that correspond to the monomeric state after a gel filtration. But in the presence of pipsqueak, it is collected in the same elution volume than the aggregated state of pipsqueak. The polycomb group complexes are known to repress the transcription of different genes, including homeotic genes, which regulate the development of anatomical structure in various organisms. And they do it through the deposition of post-translational histone modifications, which are epigenetic marks for gene silencing. So we hypothesize that the self-sustaining pipsqueak aggregates may provide an epigenetic mechanism to perpetuate the repressive state of multiple genes through generations. So this is a joint project with Kaushik-C, and now we are performing the high-resolution structural characterization of pipsqueak aggregates isolated from developing embryos. And with Javier Oroz, we are doing the monomer characterization to understand the conformational changes that are necessary to control this repressor activity in a context-dependent manner. So now the rest of the talk will be focused on the functional prion-like protein implicated in memory persistence. And here, what we want to understand is how a brief experience creates a memory that persists for months and years, even in the absence of the original experience that created that specific memory. The substrate of memory has been extensively investigated at the neuronal network level, and today, the synapse is considered as the unit of memory, which is encoded in the alteration of the synaptic strength. However, the underlying molecules and biochemical changes induced by the initial experience remains unclear. And this is what we call memory engram. A memory engram, which was introduced by Richard Simon more than 100 years ago, is a long-lasting physical substrate created by an experience that allows the storage and recall of the experience as a memory. Another interesting unknown question is how memories that can last years could be based on proteins that in average have a life of several hours in the cell. In other words, how memories are resistant to the continuous molecular turnover in the cell. So back in the 80s, Francis Crick proposed a possible solution for both questions. And basically, he said that the memory engram should be a self-renewing protein system stable against the molecular turnover. And this is basically what the definition of the prion-like proteins is, forming a self-sustaining state, as we discussed before, that can be stable on time. So during the last years, the self-sustaining aggregated state of a protein called cytoplasmic polyadenylation element binding protein has emerged as a candidate for the physical substrate of memory. The discovery of the prion-like properties of the neuronal CPEB was done by Kaushik, Eric Kandel, and Susan Lindquist almost 20 years ago. And today, there are many evidence that suggest that the aggregation of CPEB is key for memory persistence in mice and Drosophila. Indeed, the Drosophila CPEB, which is called OR2, is very well characterized at the biochemical and cellular level. And what we know is that in a non-stimulated synapse, OR2, which is an RNA binding protein, exists as a monomer bound to the 3' UTR of target mRNAs. And in addition, OR2 binds to a protein that here is represented in brown, CG13928. And this protein now recruits a repression complex, which is a deadenylation complex, and maintain these mRNAs in a translationally dormant state. Now, when the synapse is stimulated, for example, through an experience, OR2 aggregates, and now the aggregated state of OR2 has affinity for a different protein. Here is represented in orange, CG4612, and this protein now recruits an activation complex, a polyadenylation complex. And now the target mRNAs are translated efficiently. So the OR2 mRNA targets include proteins involved in synapse formation and function, which are required for memory persistence. So in Drosophila, when OR2 aggregation is abolished by specific mutations, memory fails to persist. And when OR2 aggregation is increased, this facilitates long-term memory formation, suggesting that the aggregated state of OR2 is a substrate for memory persistence. So a key question here was what structural state does OR2 adopt in the brain to function as a biochemical substrate of memory? So to characterize the function and structure of OR2 aggregates, we purified OR2 protein from the adult Drosophila melanogaster brain. And due to the low abundance, to obtain OR2 from the native environment, using no tags or overexpression systems, we used around 3 million fly heads. And using different chromatographic techniques, we obtained a 97% pure sample according with a quantitative mass spec analysis. And in this sample, OR2 is found in a monomeric state and in a SDS-resistant aggregated state. As you can visualize here, this corresponds to the monomer, and these bands here correspond with the aggregated state. And this also can be visualized in a Western blot using SDS page. This is the monomer, these are the aggregates. And also using this kind of gels, semi-denaturing detergent agar gel electrophoresis, where this large smear here correspond to the aggregated species. So the different OR2 conformational states can be separated by a gel filtration, and according with the calculated size, the different states are identified as monomer, oligomers, and aggregated OR2. So unlike in the adult nervous system, it is known that OR2 is only monomeric in the early embryo. So using a similar purification protocol, we purified embryonic O2, which remains as a monomer, as visualized by Silverstein, Western blot in SDS page, and Western blot in semi-denaturing agarose gel with no visible smear. And this was also verified after a gel filtration where the protein elutes in a volume compatible with the size of O2 monomer. So besides purify O2 monomer, this experiment is very useful for us to, as a control, to indicate that the aggregated state obtained from the adult brain is not a consequence of an artifactual aggregation of O2 monomer during our purification process. So in electron microscopy, O2 monomer was not visible, while the oligomers appeared as globular structures, globular entities, and the aggregated O2 appeared as unbranched helical filaments with dimensions in the nanometer range. And this is compatible with the approximately 1 cubic micron size of an average synapse in Drosophila. As we mentioned before, the aggregated state of O2 interacts in the synapse with two important physiological partners, the target mRNA and CG4612 protein, which finally recruits the activation complex. So in this experiment, we measured the ability of purified O2 filaments to interact with both physiological partners. And in this experiment, we labeled CG4612 with a 6 nanometer gold particle by immunolabeling, while the target mRNA was covalently labeled with nanogold particles. Both the mRNA and CG4612 colocalized in the O2 filaments, where the bigger gold particles here correspond to a CG4612 molecule, while the smaller ones correspond to mRNA molecules. And this colocalization is reduced when using mutant versions for both mRNA and CG4612 that don't efficiently interact with O2. So knowing that O2 filaments retain the interaction properties, we measured the biochemical activity using an O2-dependent in vitro translation assay. So this is a tricky experiment, and in order to do this experiment, we need 3 important components. The first one is embryo extract, which lacks O2, but contains all the necessary translation complexes. The second component is O2 purified from the brain, either monomer, oligomer, or aggregated. And finally, we used a dual reporter genetic system that contains the 3' UTR region of an mRNA target, in this case is tequila, where O2 can potentially bind and control the expression of a reporter firefly luciferase. So using these systems, while the addition of O2 monomers reduced the translation of the reporter acting as a translator repressor, oligomers and filaments obtained from brain enhance translation of the reporter acting as a translator activator, suggesting that O2 filaments purified from brain retains its functional properties. So next, we used electron cryo-microscopy to determine the atomic structure of the biochemically active O2 aggregate. And here, the vitrified individual filaments were aligned and classified using a box size long enough to cover an entire crossover. And using the homogeneous subset after the 2D classification, the crossover distance can be measured. And from this distance, we can calculate the helical twist to generate an initial 3D reference, which was further processed and refined through helical reconstruction to obtain the final 3-dimensional density map. And in this map, as you can see here, individual beta strands are stacked in perpendicular to the main axis of the filament with an interstrand distance of 4.7 angstrom, indicating the amyloid nature of the OR2 aggregate. In this animation of the electron density map, we can see the 4.7 angstrom separation between beta strands, typical of an amyloid, and the filament core has a triangular cross-section, which shows that every molecular layer is composed of 3 identical protofilaments related by a C3 symmetry. And at this resolution, a clear pattern of side chains densities can be identified. So using this map, the novo atomic model was built, and this model shows that the OR2 amyloid is formed just by 31 amino acids, and it is rich in histidine residues and glutamine residues, which form a tight interdigitated packing stabilizing the 3 protofilaments. And in addition, there is an unknown non-proteineous density running along the filament that could potentially be important for OR2 amyloid stability or amyloid formation. An interesting physicochemical feature of the OR2 amyloid is that the amyloid core is mostly hydrophilic. So as you can see in this representation, most of the amino acids that form this structure are hydrophilic and form hydrophilic interfaces between the 3 protofilaments. So the hydrophilic nature of the OR2 amyloid may have important physiological implications. So for example, if we compare OR2 with pathological amyloids, we can see that pathological amyloids form hydrophobic cores, while OR2 forms a hydrophilic core. And because of this, OR2 structure is less stable. For example, the 3-fold symmetric structure of OR2 is, at least morphologically, very similar to the structure of the amyloid beta from Alzheimer's disease. However, as shown in these solvation energy maps, the amyloid beta is stabilized mostly by hydrophobic interactions, which results in a more stable structure in comparison with the OR2 structure, which has lower stability. And as a consequence, OR2 amyloid is malleable and its stability is pH dependent. So OR2 forms stable filaments at pH 7 or 8, but changes in pH destabilize the filaments, suggesting how functional amyloids could be stable yet malleable or even reversible entities. So in summary, the OR2 monomer is a translation repressor. So before synaptic stimulation, OR2 targets, mRNAs, are maintained in a translationally dormant state by OR2 monomer. This is an AlphaFold predicted model for OR2 monomer, highlighting the amyloid forming region in yellow, orange, and green, and the RNA recognition motifs in blue. When the synapse is stimulated, OR2 forms the amyloid state driven by the prion-like domain. And now the amyloid state binds to CG4612 and enhances the translation of the previously dormant synaptic mRNAs, which include proteins involved in synapse formation and function, and therefore memory. So due to the self-sustaining properties of OR2, once the amyloid state is engaged in that specific synapse, the newly arrived target mRNAs will be activated, allowing the synapse to maintain its altered state, even in the absence of any further synaptic stimulation. So now, what can we learn from this data, from this structure? So we have shown that OR2 amyloid enhances protein translation, which is required for memory persistence. However, an open question is if the amyloid state is directly involved in memory persistence. So to answer this question, we have applied a strategy based on the discovery of new classes of functional protein aggregates, as we discussed before. And in this experiment, we have substituted the prion-like domain of the endogenous OR2 locus, which according to our structure is responsible for the amyloid formation. And this has been substituted by prion-like domains of multiple proteins that belong to different classes of aggregates using the CRISPR-Cas9 system. And now the chimeric OR2 flies were tested in their ability to produce long-term memory using a classical memory paradigm in Drosophila, which is the appetitive associative memory assay. In this assay, star flies learn to associate a specific odor with a food reward. So trained flies subsequently seek out the sugar-associated odor for several days afterwards, and this indicates that they have formed an associated memory. So what we found is that the amyloid-forming domains from canonical amyloids, like the sub-35 East amyloid, substitute OR2 function, while other amyloids from other types of aggregates don't substitute OR2 function. And this indirectly indicates that the acquisition of a static amyloid state is important for memory persistence. The next question that we are exploring is what is the molecular basis of forgetting? So in Drosophila, like in many other organisms, there is a decline in long-term memory during aging. So coming back to our model, the memory decline during aging could be potentially due to different scenarios. Like for example, changes in the structure of OR2 or in the supramolecular conformation of OR2 could lead potentially to a translationally inactive OR2 amyloid state. Or OR2 amyloid could be dissolved within time during aging, or even the memory decline during aging could be an OR2 independent mechanism. So to study this question, we first select a group of flies with different ages, from 5-day-old, which are very young flies, to 35-days-old flies, which are very old flies. And we measure their ability to produce long-term memory using the appetitive associative memory assay. And as I said before, trained flies that form an associative memory will seek out the sugar-associated odor for several days. So what we found here is that young flies from 5 to 10 days old produce robust long-term memory, while aged flies from 30 to 35 days old show an impairment in memory. And here as a control, we use a group of flies in which OR2 cannot aggregate efficiently since the prion-like domain has been deleted. So this indicates that long-term memory declines during aging in Drosophila. And this is not a new observation, but it is very useful to us to dissect group of flies with different memory index. So we selected now 3 representative groups: young flies with a good memory, aged flies with an impaired memory, and in between of both, we have the adult flies with an intermediate long-term memory. So we took these three representative groups, young, adult, and aged flies, and we purified O2 aggregates following the same purification protocol that we developed previously. And here I show the final separation between monomers and aggregates in the three groups. While monomers appears as a discrete band, aggregates show this smear pattern in the SDD agar-ox gel. And the aggregates were visualized using negative stain electron microscopy, and they appeared as unbranched helical filaments with a similar average length between the different groups, although during aging, O2 acquires distinct supramolecular conformations. So now, to gain atomic detail into these mesoscopic differences on the filaments extracted from the young, adult, and aged flies, we are performing single particle cryo-electron microscopy analysis of all of them. So next, to determine whether the reported supramolecular changes influence on the translation activity, we measured the biochemical activity of O2 filaments isolated from young, adult, and aged flies using our O2 dependent in vitro translation assay. And here, the filaments isolated from young flies enhanced the translation of the reporter, and the filaments isolated from the adult flies also increased the translation of the reporter, but not at the same level. While the filaments isolated from aged flies had a weak effect on the reporter translation. So this indicates that during aging, O2 translation activation decreases. So to understand this change in biochemical activity, we measured the ability of purified aggregates to interact with CG4612 protein, which, if you remember from the introduction, is the protein that recruits the translation activation complex. So we used an immuno-EM protocol, and we found that filaments extracted from young flies recruit CG4612 more efficiently than filaments extracted from the aged flies. And again, here, each black dot corresponds to a gold particle and represents one CG4612 protein molecule attached to one single filament. And this effect seems to be specific because the interaction decreases when we use a CG4612 mutant version that does not interact properly with O2. So after quantifying the number of gold particles attached to the filament, we can now conclude that O2 amyloid from aged flies does not efficiently recruit CG4612, while O2 amyloid from young flies does. So putting together all this data, we can start anticipating a molecular mechanism for age-related memory decline in Drosophila. And in a synapse, in the synapse of young flies, which produce robust, long-lasting memories, O2 forms a specific supramolecular amyloid conformation that enhances the translation of target mRNAs due to their ability to efficiently recruit CG4612. Now, during aging, memory declines, and in the synapse of aged flies, which show a memory impairment, O2 amyloid changes to a different supramolecular conformation that cannot enhance the translation of target mRNAs. And this is due to their weak ability to recruit CG4612. So our next goal is determining the atomic structure of the different supramolecular amyloid conformations isolated from young, adult, and aged flies to explain their differential ability to recruit CG4612. Another question that we are exploring is what cellular factor or factors drive or to supramolecular changes during aging. And here we are exploring different scenarios, like the change of ion concentration in neurons during aging due to the downregulation of specific ion channels. Or another tantalizing possibility involves the activity of chaperones. So it is known that some chaperones can be induced during aging, and also that chaperones are known to modulate amyloid disassembly. So we aim to identify chaperones that induce or to supramolecular changes that could account for the age-related memory decline. So right now we are working in these two different directions. Finally, we are also analyzing whether the amyloid-based mechanism for memory persistence is also conserved in the human nervous system. So humans and mice have 4 CPEB family members, which are homologs to our twin Drosophila, and are expressed in the mediotemporal lobe, which is an area of the brain key for episodic memory. And among them, there is some evidence that suggests that CPEB2 and CPEB3 are involved in memory persistence in mammals. However, whether the amyloid state is a substrate of memory in humans is completely unknown. So we aim here to isolate and structurally characterize CPEB2 and CPEB3 aggregates from human brains. And to do this, we have collected around 150 milliliters of dissected mediotemporal lobe from 25 to 50 years old individuals, including the temporal cortex, which is implicated in memory storage. So we have been working on this project for several years now, and our preliminary data show that CPEB, semi-purified from human brains, form SDS-resistant aggregates that display amyloid-like morphology under an electron microscope. So as a conclusion for the presentation, the understanding of the function and structure of physiological amyloids, in addition to provide insight into how transient stimuli create a persistent change in physiology in a context-dependent manner, may force us to rethink why and how other amyloids are pathological, particularly in the human nervous system, where amyloid deposition is a hallmark of neurodegenerative disorders like Alzheimer's, Parkinson, or Huntington disease, each of one affecting different neuron populations in different regions of the brain. Finally, I would like to thank all the people involved in these projects from my previous institution, the Stower Institute, and from my current institution, the University of Hong Kong, and also to my collaborators from Stowers Institute, Kausik, from the Rocasolano Institute, Douglas, Margarita Menendez, Javier Oroz, Marta Brooks, from the Cajal Institute, Mariano Carrion, from the LMB in Cambridge, Source, and Alexei Marcin, and from the University College London, Wenjuan Sang. And thank you very much for your attention.
Okay, Rubén, thank you very much for this really interesting seminar. I think it's quite stimulating because somehow connect with a capacity of humans that I don't know, makes all these investigations very valuable, because our memory is essential for us. So thank you very much. Now I think it's time for questions. So please, anybody interested in making questions can write them in the chat. And well, to begin with, I have a couple of questions. If nobody has yet written any question. So I don't know if I didn't see in your presentation, but OR2 amyloid enhances translation of what messenger RNAs? What proteins are modulated by the action of these filaments?
Yeah, this is well characterized. This is a paper from 2012 where so the targets basically are broadly classified in different groups. The first group involves genes which are involved in neuronal growth and synapse formation. The second group of genes are involved in synaptic function, and the third group of genes are involved in proteolysis, and this is quite surprising. So some of the examples of OR2 targets genes are known to be important for the stable formation of long-term memories. Like, for example, tequila. Tequila is an homologue of the human neurotrypsin, protein kinase C, which is an homologue of the mouse atypical protein kinase C. And all these proteins are very well known to be part in the establishment of long-term memories.
Okay, Douglas has a couple of questions, as expected. So thank you for this great talk, Rubén. Is it possible that some other molecule could be coding?
Some of the molecules could be coding?
Coding.
I guess he means the amyloids. It is totally possible. Yes, it is totally possible. Actually, the ideal experiment would be to try to obtain a structural information from the native environment, right? Like from the neuron. So definitely it is possible that in a more native environment, like in the neuron, the OR2 filament could be coated by mRNAs, by CG4612, or by even translation complexes. But in order to do this, we should go to the native environment, to the neuron, freeze the neurons, and try to do cryo-electron tomography, and that's pretty challenging right now. But yeah, that would be a great experiment. It's pretty challenging because there is a technical issue, and is because OR2 amyloid formation is pretty tight regulated. This means that the OR2 amyloids are not broadly distributed in all the synapses. The amyloid formation is only formed in specific synapses. So that makes the analysis very complicated for the cryo-electron tomography.
Another question by Douglas. The OR2 amyloid in the older flies to block interactions with CG4612?
We don't know what is the mechanism. What we believe is that obviously that's the main reason, because we want to obtain the atomic structure of the filament, right? To see if there is any structural feature, at least in the amyloid core, that could tell us why some filaments has better ability to interact with this protein. So what I can anticipate is that the filaments obtained from the young flies have a different architecture, are more twisted filaments, while in the aged flies, they seem like more, not totally straight, but the crossover distance is minor. So that means that there is a huge morphological change. It is possible that because the surface is changing dramatically, it is possible that C4612 maybe cannot be sterically accommodated in the aged filaments. But this is something that we don't know. We have some experiments in mind to try to prove this hypothesis, but we don't know yet.
Okay, okay. So Miguel Ángel Treviño has a question, a couple of questions, in fact. Have you tried chimeric OR2 with other more stable amyloids to check how they can influence in the forgetting process?
No, that's actually a very good, that's a very, very good experiment that we could try, but no, we haven't tried.
Could be possible to obtain flies with a longer memory in that way? Do you think so?
Technically, it could be possible. Yeah, technically, it could be possible. One experiment that we were thinking when I was in LFB, we were discussing this experiment because we know that the glutamines are very important for the amyloid formation. So we were thinking, what would happen if we expand the glutamine region and we make the amyloids more stable and longer? So potentially, it would be possible that the memory could be, I wouldn't say better, but I would say, because in order to create a memory, you have to train the animal. You have to train for several hours sometimes. So maybe having a more stable amyloid wouldn't improve the memory, but would decrease the threshold for memory formation. So these are some experiments and some ideas that we are thinking about. Yeah.
Okay. Salvador Ventura, which thank you, Salvador, for attending this talk. He's also a highly recognized researcher in this field, as you,
I know
very well. So amazing talk, Rubén. So do you expect the globular domains may interact with the fibrillary core, and may these contacts play a role in regulation?
Probably yes. We don't really have any experimental evidence for that, but I would suggest, I would say probably yes. Yeah, but we don't have data.
Sorry.
Yeah, so one experiment that we are thinking in that sense is that if you think in the amyloid, like there is an amyloid core, and then that it is coated by these RNA binding domains and everything, right? So one of the ideas that we are thinking about is try to remove all the coating using a protease like pronase. So we remove all the RNA binding domains and study the structure just of the amyloid core. And having this comparison between the full-length filament and only the amyloid core, we could try to get some information in that sense.
Okay, I say that as expected, Javier Oroz has a question for you. Awesome presentation, Rubén, thanks. Do you think one could use your atomic structure from the amyloid to fit in electron densities obtained in situ from Drosophila neurons by cryotomography?
Could be possible. It could be possible. There are several limitations. The first limitation is a technical limitation, and it's like cryo-electron tomography. For some specific complexes or protein complexes like ribosomes, you can get pretty high, actually awesome resolution. But for this kind of proteins, I would anticipate that the resolution is not going to be that good. And the second limitation is that, as I mentioned before, because it's a very tightly regulated process, the amyloid formation is specifically targeted in very few synapses for each memory. So that means that you really need to know where the amyloid is to do the cryotomography in that specific area. So that's the reason that cryotomography works very well when you have some macromolecules that are highly abundant, like ribosome, for example, right? But in this case, I think definitely would be like the golden experiment, but it's pretty challenging.
So no more questions in the chat. So I have a curiosity, Rubén. How homogeneous are amyloid or2 filaments? How homogeneous are in terms of length, of their length?
They are pretty homogeneous, actually. So when we were working with, it's a tricky question, because obviously in our purification process we are getting the protein from thousands and millions of grains, and we are passing this homogeneate through different columns. So it is possible that the filaments may be breaking, right, during the purification process. We don't really know. But what I can tell you is that after the purification process, the size is pretty homogeneous, around 75 nanometers to 150 nanometers or so, which, if you think about it, is logic in the sense that the synapses of fly are very, very tiny. So you cannot accommodate maybe micron-sized filaments. So they necessarily has to be very little filaments. So in terms of homogeneity, in terms of size, they are homogeneous. Yeah. In terms of morphology, surprisingly, most of the filaments that you can see in the micrographs are identical. So there is not a lot of heterogeneity in the sample.
Okay. So in fact, my curiosity was if you think that somehow the length of the filaments may be a parameter that somehow affect the functionality of these filaments.
Probably yes, because as we think it works, the protein is it's not that the amyloid is doing something really active. It's like a kind of a scaffold, right? So you have a very large aggregate. So obviously, if the size increases, you have more RNA binding motifs that can bind more RNAs to produce the translation of these mRNAs. So theoretically, if you increase the size of the amyloid, it should be more active in a translational way.
Okay, Rubén. No more questions in the chat, so I think it's time to thanks again your excellent presentation. And as I don't know if you know, but all speakers are awarded with a present for their participation in our session.
I didn't know.
If you come back to Spain, please come back to the institute, and this present will be waiting for you. So thank you very much.
All right. Thank you very much. Thank you very much to all of you.
Okay, and thank you all the participants and the attendants. Thank you. Bye, Rubén.
Bye.